Yeah, so as I was introduced, I am a PhD student at Southampton, and I'm trying to determine whether biotic controls buffer against climate-driven extinction threats, trying to untangle the biotic and abiotic factors of that using planktic forams. So we all know there's ongoing declines in biodiversity. There's many papers published on it. I won't go into it in detail, but we know how it's going to affect us eventually. And the main issue is how we're going to measure the loss, predict the loss in the future. So we're going to need accurate predictions of extinction risk to reduce biodiversity loss. We have evidence that climate change, predominantly temperature, is the main abiotic control of biodiversity, but identifying the biotic regulators of biodiversity change in deep time is difficult because we typically cannot observe these biotic interactions directly. It calls for long-term studies of the evolutionary response to climate change using the fossil record, but very few fossil records allow for high-resolution reconstructions of evolution. So typically, studies of diversity dynamics in deep time have only involved molecular phylogeny due to the general lack of fossil record. And this method has repeatedly been shown to contradict the fossil record anyway. So we're trying to find another way of measuring this accurately. This is where what I study comes in. So planktic foraminifera, I'm sure most of you know what they are already, but I'll do a quick overview. They're microfossils, they're unicellular protozoa, they've got calcium carbonate shells, which record the past climates because they capture the environment in which they grew, both morphologically and geochemically. So different species have different optimum environments, and environmental change will alter this morphology and geochemistry of the shells. So that's how they're so good at recording past climates, because we can measure these aspects to get those through. But this means they're vulnerable to climate change. But how are they useful for biodiversity studies? There's a very good fossil record. They are globally widespread, abundant. There's different species and assemblages found across the oceans. So, for example, the general trend is that there are more species at the tropics than there are at the poles. And within these different ocean basins, there are varying assemblage compositions, which are impacted not just by temperature, but also things such as water depth, stratification, nutrients, salinity, etc. And this variability allows us for detailed analysis of evolutionary change across different environmental settings and climate states, and it makes them a good model for understanding biodiversity. So we already know that planktic foram diversity has decreased steadily over the last 5 million years. This happened during a transition from a warm climate to one cold enough to support ice sheets at both poles. So it's widely suggested that climate played a major role in the decline in diversity, but other abiotic drivers such as ocean circulation and ocean productivity are known to have an influence. Mass extinctions have been studied extensively, but the cause of individual extinctions remains relatively unknown. But it's important to look at these background species-specific extinction events to test for threshold behaviour to climate change and assess the patterns within assemblages to see if we can use these to predict future extinction risks from climate. To explore diversity in planktic forams, we can count planktic foram species abundances to help us track assemblage changes across extinction events. This can be informative of diversity drivers when compared to environmental changes, but just looking at the number of species present isn't an adequate method to represent true biodiversity. Biodiversity is too complex to be represented by a single number, and by this I mean that, for example, if I have 2 samples and there's 30 different species in them, one sample could have for example, all of the species have the same amount in there, but the other sample could have 2 species that dominate and the rest are rare, having only 1 of each. So you're looking at 2 entirely different communities, but species richness is saying they're the same, having both 30 species each. So knowing the occurrence of each species present is therefore inherently useful too. But how can we combine these to measure biodiversity changes accurately and untangle this biotic, abiotic drivers of extinction risk? And this is what I'm hoping to do with my research. So to do this, I have material from ODP Site 925 in the Equatorial Atlantic. I have over 1,000 samples, which gives me a rough resolution of about a sample every 4.5 kiloyears. The site has good core material availability and diverse planktic foram content, making it great for tracking these extinction changes. This is due to its tropical location and it's got great preservation. According to the shipboard data, there are 28 extinctions of individual species at this site, but I've focused on 19 of these. I've discounted the more ambiguous species extinction events where there's not necessarily enough of the species in there to witness the extinction happening. And the first step of my project was to test the methodology of assemblage counts, because 300 specimens is typically used for planktic forums. So splitting down and counting 300 specimens, but this was determined using recent assemblages. But diversity was up to 1.4 times higher in the past. So I wanted to check that 300 still applied to the fossil record, particularly the time I was using, to make sure it captured species richness within a sample accurately. So I took one of my samples and did repeated multiple splits for size assemblage counts. So for example, I did 10 repeats of 10 splits and 5 repeats of 30 splits, etc. And from this curve, you can see that it sort of flattens off around the 300 mark, which means that the methodology works. I can still use 300 and accurately capture how many species are in my sample. But before I could move on to actually doing my assemblage counts, I needed to find the last occurrence for the extinctions, the species I was looking at. So to do this, I manually looked through all of the samples around the published dates from the shipboard record to find the last occurrence of the species I was looking at this higher resolution. I've plotted these here on the oxygen isotope curve, just to get a rough idea of the environment the extinction happened in. I think we have 7 interglacial, 7 glacial, and 5 transitional period extinctions. And as I was doing this, I also noted when the rough abundances changed for the species I was looking at. This is so I could establish 3 time points across the extinction event. So I have one which is pre-extinction, where the species is prevailing and doing well. I have another at imminent extinction, where the species appears to be diminishing and struggling a little bit prior to the extinction event. And then post-extinction is obviously where the species is no longer in the samples and has apparently disappeared. And from these initial counts, which is the black curve here on this graph, you can see there's a general decline in number of species over time. So we have around 37 species per sample in the older samples, and then in the younger ones, we average around 25. We do actually drop down to about 20 at about 2.25 million years, but it comes up a little bit. But when you track all of these, so I have the 3 time points for each 19 extinction events, there's no overall pattern that each of these extinctions follow when you just track it in time. So this is why we wanted to find a method that worked to track these things. But we can say that we find there's a negative correlation with oxygen isotopes, so climate is driving these extinctions. But again, like I said, there's no clear pattern, and we want to see how the other factors are playing into this. And this is where the Hill numbers come into it. So Hill numbers are used to characterize either the taxonomic, phylogenetic, or functional diversity of an assemblage by combining 3 different means of measuring diversity. So we've combined richness, evenness, and dominance here. And the method calculates diversity as a curve. Not only does this capture any individual extinctions and what happens to those species in the run-up, but it also sees what happens to the species that are left behind in terms of the abundance. So Q is the Hill number and determines the rarity of species compared to abundance, with higher values discounting all but the dominant species. Q0 is equivalent to species richness, which is what we've counted in the sample, where all species are treated equally and the abundance isn't considered. Q1 is Shannon's entropy or evenness, where abundance is considered in proportion to frequency and shows the typical species within a sample. And Q2 is the reverse of Simpson's diversity or dominance, which shows the relatively abundant species, with all but the most common being discounted. So it gets rid of all the rare species and is showing us what is dominating that community. And like I said, these plot as a curve, and you get 2 curves from this method. You have an empirical curve, which is the observed data, so basically the number I counted. Typically, empirical values underestimate species richness because there's always a chance there's some species that go undetected. Increasing the number of individuals counted increases the reliability of data, which is why I checked the methodology to make sure I was actually including them all. But proposed curves are the estimated values that reflect how many more singletons could be in the sample based on how many are actually present given the sample size. Basically, having the two curves just reduces the bias in the numbers. The slope of the curve is indicative of how even an assemblage is. So a flat curve means an even assemblage. So you've got relatively similar abundances of all species throughout. But a high gradient means an uneven assemblage where you've got one or two or three species dominating the community. And I know it's really hard to see because I have lots of plots, but this is basically all of my 3 time points across all of my 19 individual species extinctions. And there's not really any clear pattern across the extinction events when you look at it like this. But we can say that all of the empirical values are below the proposed, meaning that there are minimal differences between the two values. But we do see that at Q0, so species richness, that around 10% of the observed values sit outside the confidence bands of the empirical values, whereas all of the evenness and dominance values are within the confidence bands, which again suggests that species richness isn't entirely accurate on its own to measure diversity. But like I said, no clear pattern. We have mostly even assemblages. We see an increase in uneven assemblages in the imminent extinction period, which suggests like a disruption to assemblages before extinction. And we mostly see even assemblages again after the extinction event, with a couple that just show, if you look in the second column, a couple that show uneven. We can obviously say that all of the species diversity measures are showing lower species numbers post-extinction than prior, but it's never just by one species, it's by multiple, and it suggests a temporary disappearance of some of the other species. And you kind of saw that in my previous figure where the species richness fluctuated a little bit, although generally declining overall. The best way to view Hill numbers in this case is to plot it against oxygen isotope data. And with this, you can see a clear change across the intervals. So the pre, imminent, and post-extinction, you've got a change in relationship with oxygen isotope from a tight relationship pre-extinction to a much looser one post-extinction. And that happens across all of the Q values, so evenness, richness, and dominance. There is a significant correlation at the reference Hill numbers for evenness and dominance with climate state. So for pre and imminent extinction, there's correlation to oxygen isotope, but there isn't anything at post-extinction. But there's no significant correlation at Q0, sorry, so richness, which is what we expected because we didn't find it that accurate to begin with. But in summary, the abiotic environment structures biodiversity in the pre-extinction phase. This structuring weakens during the approach to extinction and then reduces further to become non-detectable in the immediate aftermath of species loss. So while species diversity is consistently lower post-extinction, the difference in dynamics between richness, evenness, and dominance definitely calls into question the reliance on taxon counts alone for inferring the regulators of biodiversity in deep time. And importantly, these results suggest that recovery from extinction could be controlled by niche occupation, while decoupling from climate, and thus a shift to less abiotic regulation in planktic assemblages post-extinction. And it also shows that there's recovery, not necessarily in the number of species present, but in the relationship between these abiotic and biotic factors and the number of species in the community, because every pre-extinction is correlated with oxygen, but every post isn't. So, and we tried to look into this, but we couldn't detect the change. It's either the change is happening too quickly or it's not strong enough to detect in our samples. So we're trying to figure out how we can measure that going forward. But essentially, Hill numbers are a viable method of measuring whether biotic factors buffer against climate-driven extinction. at an individual level, and it can help us untangle the abiotic biotic controls in these extinction events. And there is potential here to gain a strong understanding of how these biotic forces shaped past extinctions and use this to predict future extinction events. And I'm hopefully going to have a paper out soon. It's in write-up, so I don't know how soon that's going to be, but I'm nearly finished with it. So keep an eye out for that. But for my PhD, I'll continue to be conducting both morphological and geochemical analysis of extinct going and surviving species to see what changes are happening within the assemblages during individual extinction events at a high level, all of which will be continued to be compared to high resolution local environmental change. And hopefully by doing all of these things, I'll be able to figure out what caused the individual species extinctions in the past, see if there's any common warning signals, and whether we can use this to predict extinctions in future climates. Thank you.